The first paper is presented by Dr. Walt. The title is High Sensitivity Analysis Using Single Molecule Arrays.
Good morning. Thank you very much for this really tremendous honor. I'm both honored and humbled, particularly looking at the list of previous awardees, some of whom are in the audience today. Really just a fantastic group of people to be associated with, and as I said, it's quite humbling and a great honor. Today what I'd like to do is talk about some of the work that we've been doing in ultrasensitive detection, particularly in the area of protein detection. And we developed this method of looking at single enzyme molecules perhaps about five years ago. And in the process, we, as actually stated on the award certificate, we started looking at some fundamental enzyme mechanisms, but in the process, we challenged ourselves to trying to figure out how to use this to actually do ultrasensitive analytical chemistry. And so the outline today is going to take us through, first, a very brief description of the array format. Most of you are probably familiar with that. The second part will orient us to how we do single molecule detection in terms of actually doing ultrasensitive detection. And then the third, I'll go into some clinical applications. And the clinical applications are primarily being examined by a commercial spin-out from my laboratory, but also we have a few things we're doing in my laboratory that I want to talk about. Before I start, I do want to give credit where credit is due. All of us who are in academics who get to the point in our careers where we start receiving awards, the real giants whose shoulders we're standing on are really the students and postdocs who worked at our laboratories. This is really a list of the people who are responsible for some of the things that I'm going to be talking about today, but also I've put some other people in there that I think are of particular note because they really contributed to some of the more important things that we discovered. In particular, I want to point out both Carrie Michael, graduate student with me back in the 90s, and also Paul Pantano, who's at University of Texas at Dallas, who also was kind of one of the first people that developed these microwells. And then I also want to make sure that I thank the speakers in the session. I was selective in terms of I had to be limited in terms of who I was able to invite, but I tried to invite people from schools that I had affiliation with. The only one that doesn't fit this is Mike Saylor, who's a good friend of mine and is always doing interesting work. But Sam Thomas is a colleague of mine at Tufts, and we've enjoyed each other's company the last several years since he came there. Bob Kennedy comes from my alma mater where I did my undergraduate work, and Bob's always at the cutting edge of really exciting work. And Chris Love, who's at MIT where I did my postdoc and is working in a field that I think will resonate with some of the things that I'm talking about this morning. So bringing in two new guys, two people who have not been to Pittcon before, so this is the next generation. So let's start in with array format. And most of you who are familiar with my work have seen this slide perhaps dozens of times. This is an optical fiber array. It's about 1.5 millimeters across. It contains 60,000 individual optical fibers. We stumbled on these things back in the late 80s and really began making multiplex chemical sensors out of them in a paper that we published in 1989 in Nature. But as time went on, we realized that we needed to have much higher densities. And what we realized is that we could actually use the individual fibers to address individual elements in the array. And the way that we accomplished this was by etching the cores of the fiber, and so this is just an optical profile of a fiber optic bundle that's been etched, and you can see that in a very dilute acid, usually HCl, the glass that comprises the cores of the fibers etches fast, and the glass that comprises the clad of the fiber material etches at a slower rate, and so one gets a differential etch, and you can literally predict the rate and the depth of the wells. And so each one of the wells at the bottom face of the well is an optical fiber, which means that the contents of each of those wells is individually addressable. And so this is just an inverse plot here, and you can see very uniform volumes in these wells. So a little over 5 years ago, maybe 6 years ago now, we did an experiment. Student David Risson did an experiment in the laboratory. He was a pretty persistent graduate student, because 3 graduate students started on this, and 2 of them gave up within a week. David persisted for another few weeks and actually made this work. And what he did was he took a little stub of an optical fiber, just a couple centimeters long, that had been etched at the end, just as we had described, and he translated that through a droplet containing an enzyme, beta-galactosidase, a substrate, this fluorogenic substrate. It's just a glycoside, and the fluorophore is tied up in this glycosidic linkage, so it's non-fluorescent. And when he trapped this solution and pressed it against a PDMS membrane, all 60,000 of the wells that are at the end of the fiber, which, if you actually do the calculation, are on the order of about 30 femtoliters each of those wells gets sealed against the PDMS and fills, in the process of translating it through that droplet, with a solution of this enzyme and this fluorogenic substrate. If there's an enzyme in there, and we use this at a very low concentration of enzyme, if there's an enzyme in there, then the enzyme catalyzes the conversion of this substrate into lots of molecules of this product, and in a very short period of time you get very high local concentrations of the product, nanomolar up to micromolar, just because of the catalytic activity of this enzyme is fast. It processes things on the order of about hundreds to even 1,000 molecules per second. And so you can then localize those enzyme molecules, and this showed up quite readily. These are single enzyme molecules now having catalyzed the formation of the substrate to a fluorescent product trapped in the wells, and you can localize those enzymes. And the percentage of wells containing the enzyme correlates exactly with the concentration. And we're going to get back to this in a minute, but obviously this is a way of looking at concentration digitally. It's not a particularly useful measurement because nobody really cares about what the concentration of beta-galactosidase is. So we challenged ourselves trying to figure out how can we do this in a way that actually enables us to quantify things that we'd be interested in. And this just shows the quantification. Here's the concentration based. This is just the concentration of a half a dozen solutions. This is from the Poisson equation, what you would predict in terms of the number of active wells, and these are just 3 replicates in the average number, and there's pretty good agreement. And so we claimed in this paper that this was the first time that the concentration was measured by simply counting molecules in parallel. So now I'd like to get to really how to convert this into a practical type of measurement. And so this is just a slide that I like to show that describes the way that we traditionally make measurements, particularly things like the ELISA assays in microtiter plates, where if you have a high concentration of protein, you get a high color. If you have a low concentration of protein, you have no color. Or background levels of color. And so this is what I've dubbed analog measurements, because it's simply a way of looking at intensity and measuring concentration based on the intensity of the color that's developed. I think we'd all agree that looking down here, if you in fact could localize molecules and count them, this digital way of making a measurement would be a much more precise way of doing it, and would ultimately be the way that all of us as making analytical measurements would like to make things. It's more precise, it's perhaps more sensitive because you can localize even single molecules. So I'd like to describe how we accomplish this. But before I do, I just want to show you what the motivation is here. This is what we call the protein iceberg. And the reason it's an iceberg is that the number of known serum proteins, this is blood-borne proteins, is on the order of about 250. And that comprises all the proteins from the highest concentration protein, albumin, down to relatively low concentration proteins such as TNF-alpha. This spans 9 logs of concentration from millimolar down to single-digit picomolar. And of those 250 proteins that are known to be present in the blood, about 130 of them, approximately half of them, turn out to be of clinical utility. Okay, so that's just to give you some sense of the numbers. How many proteins are below picomolar? Well, from the human proteome, there's now probably on the order of about 22,000 genes, and if one looks at the sequences of those genes, there's probably about 1/10 of those have signal pathways that suggest that they would be secreted into the serum. And so one can imagine that this dark space, the region below the surface, is filled with all sorts of proteins that will have clinical utility. It's just we can't measure them. So how do we do it? The way that we do it is virtually identical to the way a conventional ELISA is conducted today. That is, we take magnetic beads that are coated with capture antibodies. We then create a sandwich, and the sandwich contains the protein of interest. So the capture antibodies capture the protein of interest. We then come in with a detection antibody that is biotinylated, and then we label that with a streptavidin labeled enzyme, same enzyme I showed you before, beta-galactosidase. And then if we now take that bead and do something with it, if there's an enzyme that's now attached to that bead, then the conversion of that fluorogenic substrate that I showed you earlier into a fluorescent product should be easy to visualize. And this is actually an image of one of these assays that we're calling a single molecule assay, a single molecule array, a Simoa. And you'll hear me refer to this as Simoa, akin to ELISA. It's just that it's the single molecule analog of ELISAs. So this is the way that we actually implement this. And I want to spend a few minutes talking about how we actually make these measurements and why we've developed this particular format. So when we first started out, we weren't using beads. We were just attaching the capture antibodies to the surface of the microwells. And we were just simply dipping the fiber into those microwells. And it turns out that Rob Korn said, you know, if you do the calculation here, you're fighting Langmuir. And the problem is that when you're trying to measure a concentration of 1 femtomolar, it will take you on the order of a year before that comes to equilibrium. And so we had a solution to that, and that was, let's just beat Langmuir by taking a solution, and so our protocol now is you take 100 microliters of blood, of a serum sample, you put into that serum sample 500,000 beads that contain a lawn of these capture antibodies. So let me just run some numbers for you. If you have 100 microliters and you have a 1 femtomolar concentration, which is 3 logs lower than the detection limit of ELISA, you have 60,000 molecules of that particular protein in that 100 microliters. So if you put 500,000 beads in there, you're going to get, on average, 1 of those protein molecules of interest per bead. Most of the beads are going to have nothing attached to them. Some of the beads are going to have a molecule of interest attached to them. You then go through the process of forming your sandwich using your detector antibody that has biotin on it, and then come in with streptavidin labeled beta-gal. We then take those beads, put them into the wells, and then seal them with substrate. So now every bead that has a single molecule of protein will carry with it an enzyme, and when that gets sealed, just as we did earlier, we now develop color only on the beads that have molecules present. And so this is a measurement that can be done in a ratiometric way, because we simply measure the number of beads that have signal on them after we expose them to substrate to the total number of beads that have been loaded into the well. So if you miss a few beads in wells, it doesn't matter. You just need to know how many there are. So how well does this work? So here is a comparison between an ELISA format and our Simoa format. And this is the best of all possible assays, because we're not using any capture antibodies here. We're simply using biotinylated beads, so a very simple format. Biotinylated beads, and then we're just trying to capture the enzyme label that has been streptavidinylated. Okay, so that's the best situation. We're never going to get better than this. But what you can see is that the ELISA plate begins to fall off right on the order of somewhere between 10 and 100 femtomolar down here. So that's a pretty sensitive ELISA assay, and then it hits its limit of detection. The Simoa, on the other hand, just keeps going down until you get to something like 22 yoctomoles, which corresponds to 10 molecules of streptavidin labeled beta-gal. So that's just the intrinsic sensitivity to the enzyme label, which tells you that this assay intrinsically has the ability to measure something between 4 and 5 logs more sensitive than the conventional ELISA. So before I get into the clinical applications, the other thing that we did in my group is we started getting away from these optical fibers, and we started working with these fiber optic plates, the face plates that are used oftentimes for CCD cameras to convey signal from the chip to the chip. And so what we did is we just cut that faceplate into a slide format. So this slide actually has something on the order of hundreds of millions of wells in it. And then by adapting a PDMS membrane on top that has microfluidic channels, we now have the ability to bring in solutions and fill them with beads. And the way we do this is simply by using fluorinated oil. So we will add after the beads are loaded into these wells, we'll flow a substrate through, we'll push it out with a perfluorinated oil, and then recharge it with fresh buffer if you want to make another measurement. And these are actual measurements that are made on these fiber optic faceplates. So this allows us to repeatedly load and unload using a series of magnetic switches, as well as continuous filling of substrate, oil, and then wash buffer to do consecutive readouts. So let me now transfer this. Go to the last topic, which has to do with what this technology enables us to access that hasn't been accessed before. What on that iceberg below the surface can we measure? And so in this regard, about 5 years ago, Tufts licensed the technology to a company called Quanterix. It was a startup company in Cambridge, Massachusetts. They've now moved to Lexington, Massachusetts. They have 60 employees now, and they're commercializing the Simoa technology. And let me show you the people who are involved in the work that I'm talking about. One of the things that I've found very gratifying in my career is translating technologies to the marketplace. And for those of you who are budding academics, I think there's nothing better than seeing things that you develop commercialized. And the other thing is that you realize that you just don't have the ability to muster the kind of funding or expertise in an academic laboratory that a company is able to have. So these are highly trained people and all working voraciously toward trying to make the company a success. And then these are the folks who are the clinical collaborators on some of the projects I'm going to talk about. Kai Blennow at Gothenburg, Herb Laporte, Alan Parton at NYU and Johns Hopkins on some of the work that I'm going to be talking about. So here are some assays that have been developed both by my lab as well as Quanterix. These are gold standard ELISA tests, and just to give you a sense, these are some of the sensitivity fold increases for some of these markers, and you can see that in some cases we don't do all that well. This is invariably due to a poor antibody pair. But in many cases, when you're dealing with good binding agents, we can get well in excess of 3 logs of enhanced sensitivity. And so I'm going to just talk briefly about some of these different diseases that are being pursued. And so the first one has to do with oncology, with prostate cancer surveillance. And for those of you those of you men who've reached 50, you know that once you get to be 50, every time you go to the physician, you get your PSA measured. And there's a lot of that's kind of been discredited for screening. PSA is not a very good marker, not either specific or selective. There's lots of false positives that result in biopsies that end up not turning out to be anything other than a painful experience for the individual. But what one can say is that PSA is very specific to prostate. And so for men who have prostate cancer and have their prostate removed, what happens is their PSA levels go down in the first couple of months after surgery to undetectable levels. And the undetectable level is right here. It's around 10 picograms per mil. So after surgery, within a few weeks, the levels in the blood go down below 10 picograms per mil. And these men have to come back every 6 months or so to get their blood sampled to see whether their PSA level has popped up above that detection limit, because perhaps there was residual tumor that was left in by the surgeon, or it could have metastasized before the surgery took place. And so down here you're working in the dark zone. And what this level is, the existing ultrasensitive PSA test, this is considered, anything above that is called biochemical recurrence and usually requires some kind of follow-on treatment, whether that's radiation treatment or chemotherapy. So let me now take you through the Simoa. This is the ultrasensitive PSA, 10 picograms per mil. The Simoa works in the high concentration range. There's just lots of beads lighting up. But then you get down to the low region, we expand that. You get down to the low region here, continue to expand that. And so the Simoa has an LOD of 0.01 picograms per mil, which is better than 1,000-fold more sensitive than the conventional ultrasensitive ELISA. And so the first use of the technology was actually to take a set of men whose PSA levels were all undetectable. These men, all 20 some odd men in this study, had undetectable levels of PSA. There's the cutoff right there, 10 picograms per mil. This is the standard PSA test that most of us over 50 would get. It doesn't need to be ultra-sensitive. But all these men were survivors, had no recurrence of prostate cancer after 7 years post-surgery, yet when we quantified them using the Simoa method, you can see that we can quantify every one of them. This is the LOD of the Simoa test, and so that gives you at least a lot more confidence when you get a number as opposed to just saying it's not detectable. We then proceeded to do a retrospective study. There were 11 recurring patients and 20 non-recurring patients. There had been these patients had all had 3 blood samples following surgery, and they were monitored for cancer recurrence for 5 years. And so let me just show you the results. What we did in this case is we discovered that between 3 and 6 months post-surgery, 1 blood sample in that window after surgery was sufficient to predict with 100% sensitivity and 100% specificity, the 5-year outcome of that particular patient. And so everyone here in red was greater than or equal to 3 picograms per mil, and you see that 100% of those patients recurred by 5 years. Men with less than 3 picograms per mil had 100% non-recurrence rate. So this single quantitative measurement may enable physicians to do 2 things. One is to tell these men, you don't have to worry about it. Just remove the psychological barrier. That's the preponderance of the number of patients here that are affected. But those 20% or so of the patients who recur, you may be able to intervene on an earlier basis by providing them with a regimen of chemotherapy or radiation that now knocks this down to the point where they're below 3 picograms per mil, and perhaps they will have better outcomes. Some recent work with the capsid protein of HIV. This is for blood screening applications. This is a capsid protein called P24. The Simoa that's used here is in red. You can see it's kind of right at the baseline that correlates with the nucleic acid test. It's able to detect infection before seroconversion. That means before one actually measures host-induced antibodies. And so this particular test can be conducted on the order of about 30 minutes as opposed to the nucleic acid test, which has similar sensitivity but takes 8 hours to carry out. Another study that was done in conjunction with Kai Blenau at Gothenburg, we developed some protein assays to two brain proteins, A-beta 42 and also tau protein. These are proteins that are associated with Alzheimer's disease. Our thought was we could have an early blood test for prediction of Alzheimer's. Turns out that they fail in this regard, and the reason is that both A-beta and tau protein, when you measure them in spinal fluid and you correlate that with a blood measurement, there's virtually no correlation. Presumably, it's simply a difference in the leakiness of the blood-brain barrier. But what they turn out to be useful for are for measuring brain hypoxia. These are patients who've had heart attacks, myocardial infarctions. Their heart has stopped. Their brains have gone hypoxic. And what physicians, neurologists in particular want to know is, is there brain damage associated with these patients? And oftentimes one does not know for many months if these patients are ever going to recover from a vegetative state. And again, we have the ability, by measuring the trajectory of both A-beta and tau protein in the first 96 hours after the MI, whether they're going to have a good outcome. Here you can see that the levels go up and then they level off at a relatively low level, whereas a poor outcome shows higher levels and it continues to go up for longer periods of time. These individuals are permanently in a vegetative state. These individuals restore their cognitive function within 6 months. Finally, just to show that you can use same kind of technology, we just published this in Analytical Chemistry, joint paper between my group and Quanterix. We can measure nucleic acids as well using the Simoa technology. Again, simply a sandwich kind of assay, and you get similar kinds of sensitivity down to low femtomolar concentration. Just to give you a preview of what the company is going to be introducing later this year, this is an instrument that's going to be a research instrument. It's really designed more for clinical research. It's turnkey system. 8 96-well plates will be stacked up in here, and you push a button and you'll get all samples conducted just by the push of a button in an 8-hour shift. The assay format has been migrated to a Sony disk. Sony is not making as much money as they used to on CDs and DVDs, so they're moving very aggressively into the life sciences arena, and so the Quanterix platform has 24 fluidic channels. I'm not going to call them microfluidic channels. The arrays are located at the edge of the disk here, so there's 24 channels that are simply used to load the beads, load substrate, seal with oil, and then read in the instrument. And so there's none of this. There's a lot of work on the centrifugal microfluidics. None of that's going on here. These are just being used as a convenient platform for 24 tests. Each of the tests is a 10-plex. So you can run up to 10 assays per lane and 24 samples per disk. My laboratory has been addressing two problems with this. One funded by DARPA, host immune response, to look at early infections. This just shows you the complexities of what's going on here. We've developed assays for these cytokines and immune response elements. Just very quickly, look at the bottom here. We have a 1,000-fold improvement in this assay. Same 1,000-fold improvement in TNF-alpha, interferon gamma, 100-fold improvement, and IL-2, 100-fold improvement. And so what we're hoping to do is use panels of these cytokines to be able to predict early infection in a host before the infection actually manifests itself. And then the other project funded by the Breast Cancer Research Program at DOD, our interest is really in moving from this kind of approach to breast cancer screening, mammography, which I understand is very unpleasant and has not only a false positive rate, but it really requires something on the order of about half a centimeter tumor in order to detect. And so we've been looking at some markers here that are associated with breast tissue damage, as well as some specific markers for breast cancer. And we're gearing up to screen several thousand samples over the next few months to see whether we can identify some biomarkers using this ultrasensitive test. So what I hope I've done today is to show you that you actually can do analytical chemistry by counting single molecules, and that there's a wide range of clinical applications that not only are beginning to appear, but also are on the horizon for, I think, many other kinds of indications. Again, I'd like to acknowledge all the students and postdocs and thank the committee for the honor of being selected here, and for the speakers, and also for all my friends and colleagues in the audience. Thank you.
Any questions?
This one right here.
It's obviously a fantastic story. Can you make a comment, though, about non-specificity? Where do you come from?
Raul, I think what you're asking is non-specific binding. Is that correct? Yeah. So non-specific binding actually is what ultimately determines our limit of detection, just as it does in the case of an ELISA. If you have to be 3 times the standard deviation of the background in order to make a confident call. The reason that the nonspecific binding is lower in our case is because the reagent concentrations that we need to measure single molecules are much lower. So we can use dilute reagents, so we don't have to overwhelm the surface with all these detection antibodies and the enzyme label in order to fully label things, because we only have a few thousand molecules that we're interested in detecting. And so the concentration of the reagents that we use on the back end after the capturing is much lower and results in improved LOD. So that's, I think, that's really the trick here is the ability to measure more sensitively gives us less nonspecific binding. Right now, we haven't looked at those. There are fusion proteins and things that people are looking at, and so it certainly would be worthwhile to do that, particularly now that we have a nucleic acid assay that can pick that up.
Any more questions? Thank you. Thanks.